So today I want to just give you a couple of introductory slides, one of which is to show you our website. And so I'm going to share my screen here any second now. Pretty sure you don't want to see my email, but we're going to get there. Okay, so this is the screen I want to show you. This is our website for the Centers of Excellence in Hematology. I hope you'll take an opportunity to go and look here where you can find lots of resources for your research. There are 5 different centers, and they're shown right here where you can go find out about courses that are available at those centers. One of the things I want to show you is this new link that we have, which is a services link, where you can go and find or ask questions about how do I get my cells of interest, or how do I evaluate cells. And if you click on a link like that, what you'll find is cores that can help you do analyses. So we're hoping that these things will guide people a little bit more in the cores that are available to them across the country, regardless of where you are located. And hopefully that will be supportive for you. The second piece I want to show you is this link up here that's called the funding link. And if you go in here to funding, what you will find is highlighted are a couple of individuals who are currently funded. And then if you just scroll down this page, what you'll notice is there are 2 types of applications, one which are called type A PNF, and these are essentially core credits that you can bank at any core that you would utilize. It just takes essentially a 1-page application, and the instructions can be found here. And right now we have a new call for type B PNF funding, and the due date is August 1st. This is for $75,000 for a year grant, and you can find the details of it on this website page, along with how to apply for it, the links, and where you upload your files right here. So I hope you'll take an opportunity to go look at those things. You can look here and see other individuals who've applied and successfully obtained funding from the Centers of Excellence in Hematology. And I hope that you'll take that opportunity to go look at that. So now I'm going to just take one brief minute to introduce Dr. Cancelas. He comes to us from Spain, but many years ago, where he got his MD and his Ph.D. And then he actually came, it looks like, straight to Cincinnati. Is that correct?
That's correct.
Correct, straight to Cincinnati, where he has been. He is the current director of the Hoxworth Blood Center, and he is part of the Centers of Excellence in Molecular Hematology there at Cincinnati Children's, and he is a professor of pediatrics and stem cell program leader. And we're so glad to have him present to us today. Want to let people know that Dr. Cancelas has agreed to have interruptions if you have questions during his talk. And today his talk is entitled Polarity Dependent Mechanisms of Hematopoietic Regeneration. And thank you so much for your presentation.
Thank you, Diane, and thank you to the Cooperative Centers of Excellence in Hematology for having this group of sessions. I know it's July and many people are on vacation. I can tell you I was on vacation until 2 hours ago. So, so I completely understand maybe the time of the year is not the best. But anyway, so today I'm going to share my screen. Let me see if I can get it. Yes, here it is. Share. I had a terrible, there's a huge storm here in Cincinnati. I hope that there is no problem with the power, but we had a few minutes ago a big fallout. But anyway, so I'm interested in talking to you today about our struggles with a concept that has been interesting to hematopoiesis and hematopoietic stem cells in the last 10, 20 years, and it's polarity. You know, when the word polarity comes to cells, everybody thinks of an epithelial cell. So epithelial cells have a basal part and has an apical part. And we know that inside the cells, there's a clear redistribution of different organelles and functions associated to the polarization of the cell. So when we were in the school, in high school or in college or in graduate studies, they were explaining us very well how cymogens, for instance, were really expressed in the ER, and then were moved through the Golgi, and then finally secreted in the apical part of the epithelial cell. And works very well. This is something very well described and known for many years. In 1990s, I had the pleasure to go to Rotterdam in the Netherlands, to the Erasmus University of Rotterdam. And there was a gentleman, the reason I went there was exclusively for one single reason. It was because there was a gentleman, his name was Rob Plumacher. And Rob Plumacher came to me. I was a graduate student, and this was 1995, 96, many years ago. And he told me, Jose, you know, we think that the hematopoietic stem cells really have some commonalities with other tissue cells, like epithelial cells, that people have not explored at all. And he mentioned to me some concepts, concepts that never associated with hematopoiesis. Junctions, gap junctions, adherent junctions, then, you know, precisely polarization. So these were concepts that they came to me and we were discussing. And I spent almost 2.5 years in Rotterdam, where practically every day I was discussing these concepts. So many friends of mine were telling me, you are an idiot, Jose, because how you can think that a blood cell can have anything related to junctions or polarization. I see them in the microscope, all of them look round. They have the nucleus in the center. I don't see any polarity anywhere, and I don't see any junctions anywhere, and they're floating. You know, I'm a stubborn person, and I believe in that hypothesis. And at that time is when still people were talking, you know, some people about the role of the microenvironment was unclear, the role, but there were people, many papers coming at that time saying, you know, that the microenvironment didn't play any role, that that you could do just colony-forming cell assays in order to demonstrate the functions. Of course, there was a lot of discrepancies, and many people, very smart people, were saying, yes, the stroma plays a role. And the group in Vancouver, for instance, was one of the major ones. The group in Manchester was another one. But there was a group in Minneapolis, for instance, that said, no, the microenvironment was not playing any role. Anyway, so in any event, I was very interested in trying to understand the role of stem cell function in the context of polarity, in context of the microenvironment. And that's my life. Since then, for the last 20 plus years, this is what I decided to devote my life. So I'm going to tell you a few things, and some published, some unpublished, that we have developed in the lab to try to understand the molecular insights and molecular mechanisms that really control some of these aspects, and why hematopoietic stem cells do have some specific signal pathways, that they have commonalities with polarization like epithelial cells, but also with some specific predominant pathways being activated, that they are not doing the same in epithelial cells. So it's like the hematopoietic stem cells have sequestered some of those pathways and then made them up on their own, and then do the things that they need in order to make their function, that is to make blood, right? So I'm going to stop here. Just, I don't need to explain too much to the audience today, but hematopoietic stem cells, typically you can transplant them, and I'm not going to say how, but you guys know. And initially you have 3 phases. In human transplantation, it's very clear. 3 phases. In your mouse, you can see simply the same, simply that the period is much shorter. But this typically hematopoiesis in the humans is considered, it takes around 2 years to be considered after hematopoietic stem cell transplantation. You have a steady state hematopoiesis. And during the first 30 days, 1 month, we have what we call initial engraftment phase, mostly resulting of the use of multipotential progenitors that they're able to home and engraft in the bone marrow. Then there is a phase we call an early phase reconstitution that is mostly made by late MPPs and short-term stem cells. Those are between 1 month and around 1 year, around that 1 year. And then after 1 year to 2 years is when you have what we call a steady state hematopoiesis. That is really the final record. When you have all the lineages being produced, including the lymphoid lineage, and it's stable. You don't have significant changes in the clonal development. As you know, in the last few years, there has been a lot of interest in trying to understand how this happens, because clonality and other concepts associated to clonal hematopoiesis of indeterminate potential has been shown to be associated clearly at these dynamics. And it's obvious that multiple cell populations have been associated to really the development of time kinetics of this hematopoiesis. So clinically and translationally, this is very important in many contexts, stem cell transplantation, of course, but one of them is gene therapy. Autologous gene therapy, either by gene addition or editing, as you know, not only CRISPR-Cas9 now, it's funny, you know, CRISPR-Cas9 looks like it's passé. It's, nobody cares anymore. So now you do base editing and all these things. So it's amazing, you know, how things are moving technologically in the last few years, no, 7, 8, 10 years. But anyway, so the idea is that you have a patient, you can collect bone marrow, peripheral blood, and then you can purify a population of stem cells and perimeters, CD34 positive cells, and then introduce add a lentiviral vector, or modify the genome, making a knockout or a knock-in for a gene, modifying a specific or revert a mutation, and then transplant the stem cells and be able to cure a lymphohematopoietic disease, right? This is very interesting because, you know, during that process, we know now, and we knew for many years, but now we are seeing in the patients. For many years, people thought that these things only happen in the mice and nobody cared about them. But then they realized that there's important concepts developing in the mice and validating the mice were very important for human hematopoiesis in the clinic. And it's funny, you know, it's always, you know, I'm an MD as well. So sometimes I get a little sad when I see my peer clinicians telling me, well, Jose, this is only good for mice, and who cares? In humans, I don't see that. When they say in humans, I don't see that, I get very nervous, because we don't see that because we didn't look at, or it's because it doesn't exist. And most of the times it's because we didn't look at. Anyway, so as we know very well, so there are multiple different genes that are very important in the gene repair, ATM, the P53, the PARP1 systems, and all of them are in the end controlling many downstream signals, sirtuins, PGC1 alpha, MP kinase, many of them controlling metabolism. And in fact, there's a very good connection between precisely cell division, regeneration, and metabolism. And this is, I'm not going to get into all the details, into all the specific aspects, how these are related. You guys want to ask, but I'm sure you guys know many of them. But you know, the mTOR pathway and the MP kinase pathway have been associated clearly as very important pathways associated to stem cell activity and function and survival. And the mitochondrial dependent mechanisms that they are important in the stem cell. So when mitochondria in normal stem cells, when the cells start dividing and stressed, the hematopoiesis really results in dysfunctional mitochondria. And this dysfunctional mitochondria in the end can induce to a stem cell death, or in the opposite, you can have a situation where you have development of clonal hematopoiesis associated to either epigenetic modifications and or genetics. Many people are now arguing what's first, epigenetic and then genetic, or genetic and then epigenetic, but in any event, you have final clonal hematopoiesis as a result. Another important thing is that there are distinct metabolic states in stem cells. We have what we call Roche-low, highly quiescent stem cells, that the quiescent, metabolically inactive, less mitochondria. These are the stem cells that they are the reservoir. I think the stem cells really in normal hematopoiesis, the only role they have is to be harvested for stem cell transplantation. I don't think they contribute too much to normal hematopoiesis, but in situations of stress, they become cycling and then becomes the proliferative, metabolically active with more mitochondria. The stem cells that they are probably the ones that really contribute to all the hematopoiesis during lifetime, right? So another important thing is to tell you that the stem cells have these two functions, right? On one side, they have to divide and produce daughter cells that they can maintain self-renewal, able to produce other daughter cells like them. But also they have to produce blood cells, differentiated blood cells, that they are able to maintain the mature function in the blood. And these two functions are contradictory, because if the stem cell becomes a daughter cell identical to itself, will not make blood. And if it makes blood, the stem cells will disappear and will get exhausted. So it's obvious that the stem cell has to make this decision about what to do, self-renewal or differentiation. So what we know is that this process called asymmetric co-segregation depends on polarity complex determinants, and some of them have been associated to mitophagosome and others, and there are multiple publications about exactly the mechanisms how this can happen. Still, we do not understand what really makes that these cells decide at a specific time to make a stem cell or to make a daughter cell, but we do know a few things. What we know is that these stem cells typically can return to quiescence, when divide and then become another daughter cell, and then they maintain this polarity complex determinants in a very asymmetric, so and very random distribution within the membrane, the cytosol. And this can be associated to low mitophagy, or when they get activated in cycling, they redistribute these determinants. You have increased mitochondrial activity and increased MYC activity and mitophagy. So one of the classical mechanisms that people have shown for many years that were very important in polarization and in epithelial cells have been shown very clearly is what we call polarity complexes. So the first question I had many years ago was to say, are these polarity complexes important in hematopoietic stem cells? And there were all these papers by Tanista Reya and by other people saying that, NAM was very important. And NAM is the downstream effector of these polarity complexes. As you can see here, and NAM gets phosphorated, and the segregation of NAM into the daughter cells was really marking the final outcome of the stem cell. So it can be a stem cell making another stem cell, or a stem cell making a daughter cell that was more differentiated and able to make blood. So this is what we call symmetric versus asymmetric cell division. But the way how NAM is controlled is typically controlled, and people define in many different papers, by the apical polarity complex. The apical polarity complex is a complex that is triggered by CC42 and ROGTPase. As you may know, in Cincinnati, we have had a long tradition working in GTPases. And then the part 6 and part 3 complex that control the final kinases. There are two. They call apicase zeta, lambda. This apicase zeta is in mouse, in humans called iota, but then and lambda. There are two isoforms. Zeta and iota is the same, and lambda is the other one, the isoforms. So these two kinases are really controlling the final outcome of the NAM. And this has been shown in C. elegans, in neurospheres, neuroblasts, has been shown in every other tissue where you can see an organ, and cells are highly connected within themselves. So the first question we had is this important? And then there is another complex. I'm going to tell you a little bit about that. It's called the basolateral polarity complex. And this basolateral polarity complex is controlled by another protein called Scribble. And this complex, Scribble GLDLG, was a complex very well known in epithelium, especially in the intestine, and is very important in the basolateral determination. So typically determinants that are close to the basolateral space are typically highly dependent on Scribble. So therefore the anchoring to the basal membrane in the intestine, for instance, highly dependent on Scribble function. So we also ask ourselves, what was the role of Scribble? So let me go through that. So what do we know? I'm not going to get into all the details. We published some papers on this years ago, but the first thing is just to tell you that the APKC activity is very interesting, because although APKC lambda is required for leukemia, and that's true, polarization and activity of normal hematopoietic stem cells does not depend on either APKC zeta or APKC lambda or both in vivo. This was a shock for us. In fact, it was a conundrum, because it was believed that this was a dogma. If you have a polarized cell, you need the apical polarity complex depending on these protein kinases. And we found that it's not true. So that was the first problem that we found in hematopoietic stem cells. So maybe some of my friends in 1990s were right. Hematopoietic stem cells did not need any polarity. But the problem is that when we knock out these two proteins, APKC zeta and lambda, in normal stem cells, the cells are still polarized. And we use other markers. I'm not going to get into all the details, but we saw them polarized. So therefore, that was telling us that the problem was not that the cells were not polarized, is that the cells were not needing the typical protein kinase C signaling in order to get polarized. So we did, for instance, the inducible deficiency of CC42, and these are data done by Yi Zheng, and I collaborated with him in this paper, and we found that CC42 was very important. So you knock out CC42, the stem cells lose their ability to have self-renewal, lose their ability to have symmetric cell division, and all of them get exhausted, but very fast. But this is an APKC independent pathway. So therefore, CC42 is important, but APKC lambda-zeta, no, they are not important, at least in normal stem cells. Leukemia is a different story. So if APKC lambda-zeta are not important, what are the key molecular switches transducing signal and stem cell regeneration? So we know CC42 upstream is important. What is really controlling the final outcome of the polarization of the stem cells? So let me remind you a little bit about the ROGTPases. Remember, CC42 is a ROGTPase. ROGTPases are these small GTPases with a molecular weight between 19, 21, 22. And there are three, there are right now around 30 of these small GTPases. They are from the RAS superfamily of GTPases. But I'm going to refer only to three groups, CC42, RAC, and RO. So we call it ROGTPases as a general subfamily of GTPases, and they always switch between an on and off position. So they can be on as ROGTP or RO off as GTP bound. And there are many proteins that control them. We have positive regulators, we call them GEFs, guanine nucleotide exchange factors, and negative regulators called GTPase activating proteins, GAPs. Okay, and they control many things through effectors from cytoskeletal, migration and adhesion, or proliferation and survival. We are not going to get into all the details. This is in fact a little old, but the families that I wanted to mention to you is mostly today we're going to focus on two, CC42 and RAC family. CC42 is one single protein, RAC are three, RAC1, RAC2, and RAC3. So we have published many papers. I don't know, I'm sure you guys know many of these papers, and I'm part of the group in Cincinnati. David Williams came to Cincinnati from Indianapolis. Dr. Kapoor is there in the audience, I saw him. So we joined the group in 2001, and I've been fortunate enough that we have been able to contribute in that way. So they are not strangers to regulated stem cells. We know CC42 controls self-renewal, controls home embolization as long as CC42, control CC self-renewal, proliferation, apoptosis, and aging. So we do know that these proteins are very important in hematopoiesis. However, there were some interesting things regarding polarization that we did not understand. So I was happy to have a great graduate student in my lab. His name is Jordan Alhoff. He's now in Denver, Colorado, with Craig Jordan, a postdoctoral fellow. And the question I asked him said, if APKC lambda-zeta are not the proteins that are controlling the stem cell polarization, which ones they are? He published a paper, and I'm going to tell you some of this data in the paper. So one of the things I wanted to bring first as an introduction is that to try to understand the role of the Hippo pathway. Hippo pathway, you know, is a pathway that has been well described in epithelial cells. In normal, in cancer, has been studied ad nauseam. Has been was discovered in the fly. You know, people make these interesting names to things. And the Hippo pathway is a negative regulator pathway of the JAPTAs. So here, just to give you an example, let me put here my pointer options. But you can have here, this is the MST12 LATs, and they control JAPTAs phosphorylation, and the end, the translocation to the nucleus. So in general, it's a negative regulator of this translocation from the cytosol to the nucleus. And this is what we call the Hippo pathway, the upstream, and the JAPTAs is the transcriptional factors that they are really binding of transcriptional factors controlling TET activity as transcriptional program. This is the classical pathway, has been done. The question is, and can this be affected by other systems? And one of the things that have been described in other cells, epithelial cells, was precisely the Skribble polarity complex. And we went to this Skribble polarity complex since the apical polarity complex looked like it was not important. So the first thing we did is with the bisolateral polarity complex is to see, is this Skribble and JAP1 colocalized? And we did this proximity ligation assay. For us, this is very good because it's giving us a single cell information. You know, it's very good to tell us about proteins and proteins that are close together. It provides us information on localization. So you have anatomical location, you have protein expression, not mRNA, protein expression, and you have two proteins close together in a specific location. And when stem cells, you don't have too many cells, right? So this is a very good way to do single cell analysis. So by using combinations of classical omics approaches with PLA and flow cytometry or sophisticated flow cytometry, we can learn a lot about the stem cells that we didn't know very well before. So you take wild type stem cells and you do a proximity ligation assay, you can see that Scribble and JAP1 are located in the cytosol, and they're together. You don't see anything in the nucleus, barely, maybe a small signal there. This is a negative control just to show you with negative antibodies you don't see anything. So what happens is you take these proteins called JAPTAs, the hippo pathway proteins, and you knock them down. So I talked to a friend of mine in Boston, Fernando Camargo, and he was an expert in JAPTAs in liver, and he was an expert in hematopoietic stem cells. And I told Fernando, you think that these mice will have a phenotype? And for my surprise, they told me in his lab, no, Jose, these mice don't have a phenotype. We did it, it does not. So then, you know, I'm a stubborn guy. I know Fernando think he's a great scientist. But, you know, in these things, negative data is always complicated to really interpret. So we did something very interesting. We use a model, an inducible model with poly-IC. We're continuing working with that because we found other interesting things. And then with this poly-IC, we saw that if you take poly-IC and you induce the deficiency of both JAP and TAS, and here you have the PCRs to show you how JAP1 knockout shows and the TAS knockout, there was a significant decline in the quiescent stem cells, entry and cell cycle, and this was for us very interesting. So the question is, if they are losing quiescence, these cells should not be able to survive after a challenge like fluorescein. And the answer is yes. So we saw decreasing quiescence, increasing number of colonies, so means cells are losing self-renewal activity. That's what we interpreted on our data. The only thing we can say here is that we see loss of quiescence and increased cell cycle activation on colony-forming cells. And yes, when we do 5-fluorouracil and we do serial 5-fluorouracil, the mice die of bone marrow cytopenias and aplasia much faster than the wild-type mice. So we say, okay, so if this is true, can we analyze if this TAS-JAP is they are controlling the polarization of the stem cells? And the answer is yes. So when we compare symmetric self-renewal and we use a marker, here is MYC in green color, you can see green color, and DAP is the blue to show you the nuclei. But you can see when you see the green color, and you see green color in only one cell, means that the cells had a symmetric division, means that the cell, one of the cells acquired a lot of MYC expression and activity because MYC goes to the nucleus. Meanwhile, if both are MYC expressing cells, we call it symmetric commitment, means both of them are going to differentiate into blood, into progenitors. Meanwhile, if neither of them have MYC, then you can see these cells are not going to have a symmetric division. They're going to have symmetric self-renewal, and both of them will be identical daughter cells and maintain stem cells activity. This is not only discovered by us, it's by other many people, but it's a great reporter. Now we have even a paper in revision where we show that beyond a reporter really is a MYC activity is really a major driver of the final fate of the cells. But anyway, so we see this in the reporter, and when we did TAS-JAP, we saw that then a significant change. If we look at the proportion, typically you find around 1/3 of the cells are in each of the different categories. In a normal 8-week-old black 6 mouse, you have around 33% more or less in each of the 3 phases: symmetric self-renewal, symmetric division, or symmetric differentiation commitment. However, when you knock out both TAS and JAP, the hippo pathway, you can see that there is a significant decrease in symmetric self-renewal and a significant increase in both asymmetric division and especially symmetric differentiation and commitment, indicating that these cells are now moving into commitment. They are not anymore in the self-renewing category, and they are there. So this goes very well with our 5-fluorouracil data, and we were very happy to see that we saw a beautiful correlate into loss of self-renewal activity and the final functional assistant. So the key question for us was then, okay, this TAS-JAP system has anything to do with other signaling pathways that we know. And it was the reason why I brought ROGI-TPS at the beginning is because this is where we go. So this was significantly data from our omics approach. We went to the JAP-TAS knockouts, we compared with the wild type, and we saw it's mostly ROGI-TPS signaling path. This is, as you can see here, is the P significance, the FTR, You can see mostly it's regulation, raw GTPases, small GTPases, actin cytoskeleton, and RANS, guanine nucleotide change factor activity. So all of them were the genes that we saw mostly modified. So we say, okay, we went there to see the confocal for JAP1. We saw JAP1 in the cytosol, as I mentioned to you before, not in the knockout. And then we hypothesized that in these cells, if JAP-TAS is really playing a role, as we demonstrated before, probably the core hyposignaling pathway was also being there, expressed, and should act as a negative regulator. Can we identify this is true? And if so, how does it work? So what we did is the first thing is to look at the phosphorylated form of JAP1. Remember, this is the form that typically gets translocated. And what we saw is that in the TAS JAP knockout, of course, it's gone, but yes, JAP1 is phosphorylated in stem cells as well, similar to what in other cells happen. So we hypothesized that the scribble was really controlling. So remember, the scribble was found to be bound to JAP1, so maybe JAP1 is controlling all this pathway. Scribble is controlling the pathway. And the key point for us was also very important. You know, all the functions of JAP-TAS had always been associated to nuclear activity. And you know, we thought maybe there's something wrong here. You know, JAP-TAS, the gain of function of JAP-TAS in the nucleus, of course, and it's easy to measure. You can look at transcriptomes, and you can look at TET, and you can do ChIP-seq or cut-and-run-seq, and look at the binding to the specific loci in the nucleus. I call it that easy. I mean, you do omics, and then you establish hypothesis. And it has been done by many people, so there was nothing there. But there is another topic that people have not explored very carefully, and it's the loss of JAP-TAS in the cytosol. Yeah, you gain in the nucleus, but you lose in the cytosol. Would it be possible that the loss of JAP-TAS expression in the cytosol really, you know, controls also the stem cell fate? And this is a hypothesis we tested. And this is where we use the, we started using our vector production facility. You know, this is the core that we are funded for in the NIDK grant, the Center of Excellence in Hematology. And I brought this story because it tells me, I wanted to tell you about this story because maybe some of you can get one of the small grants of $9,000 to $12,000 to use a core. And I'll be happy to tell you that, you know, it's not maybe false modesty, but we make vectors where others don't make it. So let me tell you the story. This is a protein, the human scribble. Human scribble is a big protein. Big protein. The overall lentiviral vector was a huge vector. In fact, borderline with ability to make titers. If you make a normal soup in normal conditions, conventional condition lenti, we were getting so little virus, practically nothing. We could not transduce stem cells. Remember, stem cells require a significant high MOI. So it was very hard for us to really have. So with the core and our expertise a little bit in how we optimize, we were able to make a titer, decent titer, with a really huge lentiviral vector that was able to be expressed in stem cells. And this is an example. So here you have how empty vector, this is wild type, we overexpress, and you can see this is wild type with JAP1 localized, and we see the nuclear JAP1 is reduced when we put the full length. But then when you put the knockout, and knockout here, you see there is no scribble, but then you have a scribble full length, we rescue completely the location of the membrane, or submembrane location of a scribble. We rescue also the JAP1 now in the cytosol. Remember, when we lose a scribble, we have JAP1 in the translocated to the nucleus. This is very interesting. So the scribble is really crucial to maintain JAP1 in the cytosol. When we put a scribble, that big plasmid, we're able to maintain the JAP1 in the cytosol. So we were rescuing the similar situation as we had in wild type or even overexpressive cells with high levels of scribble. But the funny thing is that we use structure function mutants. This is what easier to express because they are smaller, because we lost part of the cDNA, of course, and we use either the amino terminus, we call them the leucine-rich repeats, LRR, and when you see in the LRR, you can see also location of the protein cytosolic, but we don't rescue the JAP nuclear translocation. When we put the PDC domain and 1,4, we can see the protein, but we do rescue. Then you can see that they are in the cytosol. So what's telling us something very interesting is that the two parts of the protein, the amino terminus and the C terminus, were really controlling two different functions. So meanwhile, the amino terminus was not able to rescue, the C terminus was rescue the JAP translocation.
Can I ask a question of that?
Sure, of course. Yeah,
Go
sorry.
back. The puncta
Yeah, let's go back.
The puncta on the plasma membrane, what
Yeah.
is that? Are
So-
those, or are they organelles? Are they
you-
vesicles?
No, this is location. So what we noticed, of course, depends on the protein expression, right? But we found, of course, there are some areas that you have more accumulation. What we have identified is that there are some specific locations, and probably we associated initially to location within the microenvironment. So we thought it could be focal adhesion kinase locations. We think it's cytoskeleton dependent. We think that these are specific areas where we have some significant increase in actomyosin and lipid rafts. We don't know very well the composition of this punte. That's a very good question. We do think that it's highly dependent of the cytoskeleton, and this is something that we are in a different project looking at, how these punte are really being controlled by a scribble in the context of hematopoietic stem cells.
Mhm.
I don't know if that answers your question.
Okay, yeah, thank you.
No problem. But that's a good observation. That's very good. So the thing is that we also look at LATs. You know, LATs is the upstream negative regulator. And what we found is that you have these scribble knockouts, LATs is gone. If you put the leucine-rich domain, LATs is coming back. So that was telling us that the leucine-rich domain was crucial to control LATs location in the membrane, on the site, in the cytosol, just submembrane. Meanwhile, JAPTAS was controlling precisely the is binding to PDC binding domains. So together was telling us that these two parts of the scribble protein really was working as a scaffold. And in one side, the amino terminus was controlling the location of LATs, and the PDC binding domains, the location of JAPTAS. So these three proteins were together, really making a functional complex that was in the end controlling some cytosolic functions associated to JAPTAS. When these proteins, in this case, scribble is lost, you lose the LATS1-2 negative regulation, JAPTAS gets phosphorylated and translocated to the nucleus, and that's the way how it works. So we look at the scribble knockout and TAS-JAP knockouts, and we found around 650 genes that were in common between the scribble knockout and TAS-JAP. I didn't bring all the data about the scribble knockouts because it would take me some time, but I will tell you in a second. So what we found is mostly GTPase activity. GTPase activity was the most important thing that we found there. And what we found is that GTPase activity was associated mostly to RAC-specific guanine nucleotide change factors and CDC42 specific guanine nucleotide change factors. So for us, this was telling us there was something important that the scribble knockout and the TAS-JAP knockout were really sharing, and it was precisely differential regulation. In this case, TAS-JAP was upregulation of RAC and CC42 specific gaps. So we look at precisely JAP with CC42 because this was the most important specific gaps that we found, CC42, and we look at that they are together. So JAP1 and CC42 are in the nucleus. So therefore, now we found the system that really works in the cytosol. It's not APKCs, it's not a typical protein kinase lambda and zeta. It's just the scribble JAP1 TAS complex. We found in the wild type, and we have even antibodies that can recognize the activated form of CC42 bound to GTP. We found the same as the total CC42. And when you look at the frequency of stem cells with proximity ligation, you can see that the scribble knockout has a significant decrease in the ones where you have copolarization. So we found not only that there was the proteins are together, but when we look at the scribble knockouts, and here's just to show you examples, and of course you can find the three groups of cells with no signal, signals that they are non-polar, and signals that they are copolar, where both proteins are together but polarized. And you can see is that when you lose a scribble, the cellulars that they are copolar, where you find both JAP1 and CC42 significantly. So that was telling us that these distinct polarized states of stem cells that we find as these three, with the ones with no signal, the ones with non-polar, and the copolar, were really distributed different way when we had wild-type stem cells or we had the scribble knockout. Meanwhile, in wild-type stem cells, 75% of the cells had copolarization. In the scribble knockout, only 30% of them were having copolarization. So the scribble knockouts, just to give you an idea, because the scribble knockouts are very interesting in mice because they have precisely the opposite phenotype as the TAS-JAP. So indicating that scribble probably is really controlled as a negative regulator. And this, when we lose a scribble, we lose the negative regulator and we have this exhaustion. When you lose the scribble knockout, one of the key questions that we found is that a scribble knockout have, in the short term, have a decrease in the competitive red population activity when you do competitive serial competitive red population assays. Secondly, you see practically the same, but when you do tertiary transplants, you can see that the scribble knockout stem cells win the competition. You have more scribble knockout gain of function of activity in the tertiary transplants, indicating that the self-renewal capacity of the scribble knockout cells are really maintained. So we did the same experiment as before we did the TASYAP knockouts, but now with the scribble, and we found the opposite. Meanwhile, around 1/3 are in each of the populations for the wild-type stem cells. Now, symmetric self-renewal represents 50% of the stem cells. You gain self-renewal. So it's precisely the opposite phenotype. So we have to precisely, and they are in that depots in the way of these proteins that they are, remember, together in the cytosol. We did paired cell assays. I'm not going to get into all the details how to do this assay. What we found is that in the paired cell assay, the scribble knockout cells, you lose the cells that have the symmetric differentiation and the asymmetric division, but you maintain, or even leave it expand, but mostly maintain symmetric self-renewal stem cells. So what the scribble deficiency does is not so much that expands tremendously the symmetric self-renewal, is that induces the loss of the symmetric differentiated cells and the asymmetric dividing cells. So you see only the ones that have self-renewal capacity. We did apoptosis, and we found the apoptosis is increasing the scribble knockout stem cells. And we found that when you reintroduce a scribble, you are able to rescue this in the scribble knockout. And especially you rescue this when you put the leucine, the PDC domain, the one that controls JAP. So it was telling us that this enrichment on symmetric self-renewal is highly dependent on the binding of the scribble to JAP. That is the one binding to PDC domains in the C-terminus part of the protein. So we did the final experiment. What happens if you knock out everything? You knock out the scribble, TAS, and JAP. And this is an example with the triple knockouts. So these are mice that have 4 different transgenes, MH1, CRE, and then scribble, TAS, and JAP, flux. And then what you see is that you can rescue the colonies. You see TAS, JAP has a significant increase in the colonies. When you do a scribble, TAS, JAP, you got practically the same values as the normal. You can rescue the cell cycle entry. So remember, when you have a TAS, JAP knockout, you have a loss of the quiescence, but then you can rescue at least partially the quiescence in the triple knockout cells. You rescue significantly the loss of the cell cycle entry when you have the triple knockouts. And when you put 5-fluorouracil, you rescue the mice from 5-fluorouracil response. So mice that they are triple knockout, now they have completely lost their ability to control the polarity system, the scribble, TAS, JAP. But interestingly, now they don't care. They are able to go into symmetry. It's obvious that they are using an alternative path. And this was our question. If they don't use the CC42 scribble, JAP, TAS pathway for polarization, but the cells now are rescued functionally from a cell cycle entry and 5-fluorouracil response, what is it? And then this is what we did this experiment. So let me tell you more. So we look for confocal microscopy, CC42 polarization, MFI, and polarization were fine. We look at CC42, GTP, but then what we found is that the triple knockout has even less CC42 activity in those cells. Again, so you don't have CC42 activity. What is the problem? How these cells are able to function normally and make the hematopoiesis be fine and respond well to 5-fluorouracil. What we found is that is RAC. So the cells now decide to go to an emergency program. They lose the normal conventional program, CC42, scribble, JAP, TAS, and then when they lose everything, scribble, TAS, JAP, activate RAC1. You can see here that the scribble knockouts have decrease in RAC1 activity, TAS, JAP have more RAC1 activity. When you have the triple knockout, it's very clear. You have a ton of RAC1 activity in that RAC1 GTP pull down assay. So, and then it's more. When we look at the GEFs activation, now they move mostly to activation of the RAC GEFs, the RAC1 nucleotide factor. So the cells are changing the pathway. They are changing the pathway. I can tell at this point in time, we are now looking specifically at the mechanisms, how this works, because now we do believe that this is dependent on the TAS-JAP nuclear activation. So remember, we talk about the cytosol loss of cytosol activity, but nuclear activation has been shown. When we look at the nuclear activation, we look at TET pathways and TET associated, and we never found them. That's the reason nobody cared about TAS-JAP, because nobody found the classical downstream targets associated to TET activation. And we don't find the TET activation with the classical TET transcriptional targets. But what people forget is that there are alternative systems of transactivation induced with JAP-TAS that they do not depend on TET. And they have been associated with ranks and many other programs. And now we are working precisely in trying to define that. In fact, we have now data indicated that probably this is related to a hypoxia-inducing factor 1 alpha in the nucleus. And this is something we are working intensively right now, and try to define a transcriptional program that had not been defined before associated to JAP-TAS. So, and also this case was good for us because increased RAC1 levels were reported to increase stem cell repopulating capacity both in vitro and vivo, and this was the data coming from UCLA from the group of Kwarmin et al. So this is more or less the summary of the talk, and I'm going to stop here. I have another story, but I don't have time. We found this complex, Scribble CC42 LATS1 JAP12, that is in the cytosol activated by a GEF, the cytosol of hematopoietic stem cells. And this complex is crucial to cell fate for self-renewal versus asymmetric cell division. And when this system is in place, you have normal balance between G0 stem cells and cyclin stem cells. They respond to upstream mechanisms that we do not understand yet. And this is part of what you were telling me before, exactly what lipid rafts and what signaling pathways they are being activating a Scribble dependent pathway. But what we know is that we lose CC42, we lose stem cell fitness. When we lose JAP1-TAS, we lose stem cell self-renewal. When we lost Scribble, we have increased stem cell self-renewal. And when we lost Scribble JAP1-TAS, we restore stem cell fitness because of RAC activation. So cells go to a plan B. This is the plan B. Now it's very famous on TV all the time for other reasons. So I'm going to stop here just to, if you can allow me to go to the last slide to tell you the important people. These are the people who have helped us a lot, have done all the work. Especially I want to mention Ramesh Nayak and Jordan Alhoff, have been working very intensively. Abhishek Singh is catching now in this study. Shala Yahechte, who is very interested in the alternative pathways now with the Scribble. And of course, our collaborator, good friend, Isaac, Teodosia Kalfa, Marie Dominique, who has been very important to us in all this study, and our collaborators in Israel, Esbela Pirot and Karim Golan. And we published a few papers with them. And some of the people who gave us some money, including the Nadike Center of Excellence in Hematology. And I'm going to stop there. I'll be happy to answer any questions that anybody can have.
Great, thank you. Feel free to unmute yourself and ask anybody.
I see Dr. Kapoor there. He wants to ask questions.
Hi, Jose. How are you?
How are you doing, Ruben?
That's great. Outstanding talk. So this seems like, so you've got reduced activity of CDC42 and increased activity of RAC. If you put the RAC inhibitor in those cells, then what happens to polarity? And secondly, in those mice, what is the engraftment potential of those cells? I mean, because you've got one GTPase antagonizing the other GTPase's activity.
So these are very good two questions. So let's go one by one because they are very different ones. Regarding the second, it's easier for you to tell you, and we have not done the transplant of the triple knockout. So we did 5-fluorouracil, and this is the data I show you. We have not done the transplant. This was at the end of his stay in my lab, and we decided not to do the transplants because, you know, in order to do the experiment well, we would have done to do primary, secondary, and tertiary transplants in order to identify if we do a phenocopy or not, and we were able to rescue the Scribble or not. Okay? So that was a lot of work, and we decided not to do it. But the NSC23766, or another one, for instance, the one you use, a RAC inhibitor, that was a good idea. And in fact, we did a small experiment. We didn't finish those experiments at this point in time, so I'm not ready to show you the data, but we did a small interesting experiment. And the funny, interesting thing is that for first time, we see the cells that they lose complete depolarization. So, of course, polarization we define as tubulin polarization, but we lose the tubulin, at least significantly tubulin polarization is decreased. So we do think that after RAC, there's nothing else. So we think it's CC42 and RAC. There is no RO or other GTPase or any other thing, but RAC is the plan B. Cells have RAC, and I think RAC is helping them when they lose everything else. So, but physiologically, the complex that they like is this complex, the CC42 Esquivel Yaptas.
Right. Thanks.
Any other questions?
I actually have a question. That was a really wonderful talk and really exciting. And I wish we could have seen the rest of the work, but I can tell that it's it'll probably continue to be published very soon. So I was actually wondering about the CDC42 in the mutants. You showed that it's very low. Is it just not being transcribed or translated, or is this protein actually being degraded? Like, is there some mechanism that's turned on, either in the context of culturing stress that might be different from in vivo, or, I'm just curious about, the cellular.
This is a very good question. So we think it's a little of everything.
Okay.
So first of all, the transcript is a little decreased, although not too much. The protein is decreased and the activity is decreased. And we think that is really an amplification cascade. Now, what are the mechanisms that are controlling the protein expression? If it's degradation, we think it's mostly about degradation than really protein expression. mRNA is not, it's decreased a little, but we have not done the formal experiments to tell you what's the major reason for that. We think it's most likely degradation, but this, I can tell you, I don't know for sure, because we think that CC42 without the rest of the complex is probably more unstable, but I don't know. As you know, many of these GTPases, only in the last few years, we have identified many post-translational modifications, including ubiquitination. Before, we didn't even know about the fate of these proteins. Now we know that these proteins also, like other proteins, get ubiquitinated and get degraded. We have not looked at ubiquitination CC42. That's a very good question, but it's possible that there is more degradation. We see less activity, and activity is easier to explain, but the protein expression, protein degradation, we need to do those experiments.
Yeah, I was actually curious about the RAC1 as well, because there's a certain fraction of RAC1 that can exist in the membrane and, tethered to different microdomains as well. Have you looked at any different, fractions of that protein as well?
No,
whether there's,
we did some lipid rafts experiments. I can tell you, I'll be honest, we didn't finish those experiments well. Those are tedious experiments. And I cannot give you a conclusion on that. But we know RAC1, you're right, is bound through the prenylation site to the membrane, a fraction of that, and it's bound to many other things as well. They like to bind to membranes, can be inside the cell or in the plasma. But we have not, I cannot provide you more information about quantitatively how much location of the RAC1 is there in those cells. Yeah, but that's a very good question.
Very cool. Thank you so much.
Maybe I get another postdoc, maybe I can continue with
I'd be happy to chat about it. I have my own lab, but I think this is
okay.
Very cool.
I know. They're not talking about you, but somebody knows somebody who's looking for a lab. Please let me know. Okay. Any other questions?
I think that looks like that's it. I want to remind people that this is recorded and will be available on the website. So maybe those who are on vacation will have a chance to hear the talk as well. It should be posted in the next couple of days. And if you go to the CCEH.io website, you should be able to click on the links to be able to find the recordings that are available there that have been recorded, and also this current one that we were just listening to. So thank you again very much, Jose, for your presentation. Take care, everyone, and see you again soon.
Thank you. Thank you, everybody.
Okay. Bye-bye.
Take care. Bye.